In this video, we'll discuss about the histopathology of dentinal caries. So, first of all, we'll discuss the early dentinal caries. Now, in this, there are five zones. We will discuss each of them one by one. So, starting from the first zone, which is the zone of fatty degeneration of odontoblast process. Disposition of fat globules. It precedes the sclerotic uh, phase. So the sclerotic phase is going to be the second one. What is the significance? The fat, it is responsible for increasing the impermeability. So fat contributes to impermeability. It is a predisposing factor for sclerosis. Also, what we will see is that the initial decalcification, it will start from the dentinal tubules, basically of dentine. It will involve the walls, allowing to distend for the microorganisms to get packed. Now, this gives a beaded appearance. Then now we will discuss the second zone, which is the sclerotic dentine zone. Now, why is sclerotic dentine actually formed? It is formed because of the reaction of the vital pulp. It is formed in a response of reparative manifestation. Basically, what it is trying to do is it is trying to seal off the dentinal tubules so that there is no further penetration of the microorganisms. Now, in this, it will be very prominent in slow caries, slowly advancing caries, and in rapidly, it will be minimal. Now, it appears as white in transmitted light. Now, the third zone that we will discuss about is the decalcification. Decalcification. Now over here of dentinal tubules. Now here what you will see is the initial decalcification. It occurs on the walls of tubules, whereas the later calcification, it occurs on the entire thing. It advances. All right, after this, the fourth one is of microorganism invasion. Now these bacteria are called as pioneer bacteria. Okay, now what happens is when it is initial phase, so we will see that there are acidogenic bacteria, whereas in the later stages we will see that it is proteolysis. Now there was a theory which was given by it was given, and it said that the proteolysis is the first one, but it is not proved according to the gnottobiotic theory. It was the proteolytic theory. Later it was seen that acidogenic is responsible for the initial stages of the dental caries, whereas the proteolytic enzyme microorganisms are responsible for the advancement of the dental caries. So this is it. And now the fifth one is the one which includes the destruction. So basically the destruction of dentine, or the decomposed dentine. Now we discussed about the early. Now we will discuss about the late changes, late dentinal changes. So basically you can draw something like this. All right, now over here, first of all, what we will mention is the decalcification of dentinal tubules. Then you will see that there is a focal coalescence of adjacent tubules and ovoid area of destruction. This is the liquefaction foci. Now, then there will be thickening of the sheath of Newman along its course. Then we will see that there will be formation of transverse clefts. Now, in initial decalcification, we will get to see acidogenic organisms, and in the later destruction of matrix, we will see proteolytic organisms. Then talking about, there will be necrotic area of dentine, which will have a leathery consistency. Then there will be an increase in the diameter of dentinal tubules to accommodate more microorganisms. Also, what we will see is that there will be peeling away of carious lesion. Then we will see that it will extend at 90° to dentinal tubule and parallel contour lines. Then there will be multiple areas of destruction. So this completes the histopathology of dentinal caries. For the diagram, you can just draw the liquefaction foci of Miller. Out of these two diagrams, you can draw any of them. So this completes it. Thanks for watching.